we're kind of making all this happen, right? You guys are sort of the unsung heroes, doing all this stuff, the support stuff, so that we can get funds and allowing us just to think about the science, and that's very helpful, because I know a lot of my colleagues around the country and around the world, they don't get to do that. They have to do a lot of the nitty-gritty stuff that goes into, you know, funding a lab and doing that stuff. So we're very appreciative of what you guys do. As Rob said, I have a couple of sort of scientific irons in the fire. I'm just going to talk about one of them today. Some of the other ones as a lab, we're interested in the mechanisms by which blast, like explosions, like IEDs and stuff that soldiers would experience, how that causes damage to the auditory and vestibular or balance system, and what we might do to mitigate against that kind of stuff. The lab also works closely with faculty in the otolaryngology department doing translational research, thinking about how we can design better kind of hearing prosthetic devices such as cochlear implants or different types of hearing aids. I'm not going to talk about that today. I'm going to talk about some recent work that we just finished up. So it's sort of the title of this is Nature or Nurture, the role of experience in the development of spatial hearing. And I'll sort of say what I mean by spatial hearing. But of course, I don't do all these experiments. I've been blessed to have kind of wonderful staff, students and postdocs and whatnot. A lot of the work I'm going to talk about today was done by Kelsey Anbuel and Alex Ferber, two graduate students who defended, you know, literally just a few months ago. Alex Ferber is an MD PhD student, so he's recently kind of cycled back into his MD portion of his training, so he's still on campus. Kelsey recently just left and is now at New York University doing a postdoc. A couple other major players, Nate Green, Andrew Brown, a postdoc who unfortunately is leaving the lab in a couple weeks and just got a position at the University of Washington in Seattle. Victor Beneshu was a postdoc for a while, is now back in Paris. And Renee Banakis-Hartle is a otolaryngology resident who spent 2 years in the lab, protected research time as part of a T32 training grant that we have in otolaryngology. But again, Kelsey and Alex's work are sort of the primary stuff I'm going to talk about today, so it's sort of fresh in our mind.
It's a great picture, Victor, by the way.
It's a classic picture. Yeah. Something's got cut off here, but basically up here what this says is critical periods for sensory development. What do I mean by that? So we believe that in human and other animal development that there's this kind of early period, you know, shortly after birth where there's a period of increased plasticity where sensory input and also motor skills and things like that, but sensory input can sculpt neural circuits and behavior. And there's a few sort of kind of famous textbooky examples of this. One is this notion of imprinting, right? So if you have some animals or ducks or geese or something kind of early in development, you know, they really sort of latch on to kind of whoever's sort of taking care of them, right? So there's these classic, you know, images of ducks following people around, things like that, right? And this happens sort of early in development and not later, right? So there's something special about early development. Another kind of famous neuroscience example is bird song learning, right? It's sort of a tractable kind of experimental model for how we as humans learn a pretty complex language. Birds also, of course, songbirds have a really complex language that they really learn to use their song early in development, and they can't do that later. Okay? So there's something special. And one of the most famous examples is our development of what we call binocular vision, right? The way we use our two eyes to have things like depth perception. And two famous neuroscientists, the last names of Hubel and Wiesel, actually won the Nobel Prize in 1980 for their work on basically the sensitive or critical periods in the development of our binocular vision. And just as an inroad, because my lab is basically stealing or borrowing from what these Nobel Prize laureates did in their work. So let me just step you through what they did and what they thought about. So this sort of classical critical period for binocular vision development. Basically, here's how we use our two eyes. So basically, when we fixate an object in our environment at some distance, we want that object to fall on a part of the back of the eye called the fovea. And the fovea is the place where the photoreceptors that basically transduce visual energy into kind of neural activity. The fovea is just packed really densely with these photoreceptors, right? And it gives us very high visual acuity so that we can do things like thread a needle and stuff like So basically, when we fixate an object in the environment, we're placing our eyeballs such that that object that we're fixating, this green object here, falls upon the fovea. Okay? So then how do we tell whether objects are closer to or further than that point of fixation? So if we have this blue object here that's closer to our point of fixation, that object falls on a slightly different part of the retina, as you can see here. And objects that are further, you can see, fall on kind of the other side of the fovea. So we call this retinal disparity. It's basically the spatial disparity between the fovea and where this other object falls. I know that's a little bit complicated, but basically we have neurons in our visual cortex that are very sensitive to this disparity. And it's effectively those neurons that are integrating inputs from the two eyes that tell us whether an object is further away or closer to the point of fixation. And it had been known for some time that kids, that had like cataracts or strabismus is common. So when one eye kind of points, deviates, even very so slightly. So actually, my daughter was very slightly strabismic. One eye, we couldn't even see it. But in kind of routine kind of vision exams, the vision acuity was getting kind of worse and worse in one eye. So luckily the doc's here, at CU found that. And then what my daughter had to do to fix that is you've probably seen many kids wear an eye patch, right? Mm-hmm. And this sort of wearing of the eye patch over the good eye, right? You're basically patching the normal seeing eye, allowing that other eye to strengthen these neural connections. That all came from this really basic fundamental neuroscientific research that I'll talk about. Somebody have a question? So this is the experiment that Hubel and Wiesel did, is to basically have a kind of an animal model of what the clinicians were observing in the clinic. That these kids that were slightly strabismic or maybe had a cataract in one eye and things like that never really developed this ability to use both eyes. I don't know where I got this picture, but Kelsey probably found it somewhere. So essentially what Hubel and Wiesel did is to simulate kind of what happens in these kids, but in an animal model where we can then understand the changes in the anatomy and the physiology and behavior. So what they did is rear cats, kittens with essentially a patch over one eye. And what they found is that if they then looked into the visual cortical neurons in normal seeing cats, most of the neurons in the visual cortex are what we call binocular. So the neurons are responsive to inputs from both eyes, right? Essentially, they're integrating inputs from both eyes. So you have this sort of bell-shaped curve. The center of this curve indicates neurons that are sensitive to both eyes, and the kind of tails of the curve are sensitive to one eye or the other. But after this period of actually pretty brief, what we call monocular deprivation, so closing one eye, if you then took the patch off, basically all of the neurons only responded to the eye that was kind of experiencing the world. There was no neurons that were sensitive to information from both eyes, and so these animals basically had no binocular or stereoscopic vision. And then what they did show is that there was a particular period pretty early in development where you had to have normal inputs to both eyes for these circuits to develop. And so if you put a patch on, for example, in a juvenile or adult animal, it didn't do anything. You take the patch off and everything's back to normal. So they really demonstrated this early period of plasticity that was apparent in binocular vision. You need normal patterned visual input during this early period, and anything that disrupts that causes a change in the anatomy, also the physiology, and ultimately behavior of the animals. Right? And so the question is, well, why might there need to be a period of a critical period for the development of our binocular vision? Why can't you just sort of wire it up? This would be the kind of genetic argument, right? That the genetics can basically wire up the system by itself. And the issue here is that, well, basically from birth to about our teenage years, the human head basically doubles in size. Okay, and this creates real problems for this notion of this binocular disparity, right? This difference between where objects fall relative to our point of fixation. So if we double the size of the head, the eyes move further apart in space. Okay, so that means this for a given object distance, that's going to have a different, basically, neural signature in a baby than it would in a juvenile or adult human, because the eyes move twice as far apart. So interestingly, this period of, or at least where the clinicians think this sensitive period in humans for development of binocular sensitivity is about sort of 5 to 7 years of age, and that's roughly the time where the head becomes almost fully grown. There is some growth up through the teenage years, but it's really not very much, right? So there's this nice correspondence between the head growth and this critical period. And we've tried to make the same argument in our work that moving on to so that sort of sets the background. So why do we think there might be a sensitive period for the development of our auditory system, particularly our spatial auditory system? So the mechanism by which we're able to know where a sound is in space. And in order to do that, we use two pieces of information. So just like our eyeballs get different pieces of information from the environment, our ears are kind of physically located at different points in space, right? They're separated by the head. So they're getting slightly different input for a given sound source. So one of the pieces of acoustic information that we use to determine where a sound is in space is the so-called interaural time difference. So if we have a sound here off to the right, that sound arrives at the near ear a short time before it arrives at the far ear. So there's a slight timing difference between the two ears that we can use to determine kind of where in azimuth, or the horizontal location of the sound is. The other piece of acoustic information we use is the so-called interaural level difference. So the head essentially acts as an obstacle to the propagating sound. And so the sound pressure on the far side of the head is less than at the near side of the head. The head essentially casts what we call an acoustic shadow, not unlike an object. If you shine a light on it, casts a shadow, right? It's the same principle as sound. So we have these two pieces of information, and these are going to be, these are sort of physical cues that are dictated by the size of the head. So in an infant, these interaural time difference and interaural level difference cues are pretty small physically in terms of their magnitude, because the size of the head is small. But as we grow, our head doubles in size, so these acoustical cues basically double in magnitude. So we think, again, over this sort of 5 to 7 year period of age. So we think this is a sort of rationale for why there might be a sensitive period. So we wanted to investigate this scientifically. And there's a couple of reasons why. So again, here's my daughter, my older daughter, Lucy, as her head doubles, she thinks her head's bigger than it actually is. But, is there a sensitive period going from infancy through kind of juvenile? And this sort of gets at this notion of why might, it's what I tell my students is it's the sort of who cares question, right? Can you explain to your grandmother why the NIH should give us money and these kind of things? Why should we care if there's a sensitive period? So let me just back up and say what a couple of the sort of primary functions of the auditory system. I've talked a little bit about sound localization, so we can determine where a sound is in space. And of course, probably more important, we can determine what it was in the environment that produced that sound, right? Is it this phone, or is it the speech from this talker? And actually our ability to determine where a sound is actually facilitates our ability to identify what that sound is. One of the kind of common things we often talk about is the cocktail party effect. So you're at a cocktail party, you can carry on a conversation with somebody, even though there's lots of other conversations going on. And the way we're able to do that is by leveraging our sound localization system. So by knowing kind of where somebody talking is relative to other sound sources, we're able to sort of focus our attention on that source and squelch out everything else. So it requires a pretty functional kind of two-eared system to be able to do that. So these two basic auditory functions sort of have an interplay with each other that I think is important. A second thing that sort of motivates us is, again, that a very brief even temporary hearing loss early in development can have really long-lasting effects that persist beyond the hearing loss. And one of the most common ones that we study is essentially ear infection. So virtually all kids have ear infection. So an ear infection, basically there's an irritation in the middle ear here. And what can happen is that irritation can cause fluid to build up in the middle ear. And you've probably all experienced this. It's like having water in your ear. If you have fluid in the middle ear, we have what's called a conductive hearing loss. It has nothing to do with the nervous system. It's basically a mechanical type of hearing loss. This fluid doesn't allow sort of the sound waves to be transmitted through the middle ear, through these tiny little ear bones into the inner ear for neural processing. So fluid accumulation in the inner ear can cause about a 30 decibel hearing loss. That number doesn't mean anything to you, but 30 decibels is about what kind of foam earplugs do, or if you put your finger in your ear, that's about 30 decibels of hearing loss. So, of course, ear infections are very common in kids, and for reasons we don't understand, about 15 to 20% of kids essentially have ear infections all the time during development, kind of where we think these critical periods occur. And so that's changing, or basically causing fluctuating hearing loss into the inputs to the system. Again, appealing to the visual system, one way I like to think about this is it's sort of like a cataract in vision. So it's kind of a blurring, if you will, of our acoustic input. And we know from these classic binocular vision experiments that even a little bit of a cataract can cause basically long-term processing problems in terms of the integration of inputs from the two eyes. So we think there's something similar like that. And we know from the literature, from kind of longitudinal studies in kids, that you can have long-term speech and language deficits, even from a pretty temporary hearing loss due to an ear infection. And another interesting thing is that these kids often have real difficulty in localizing sounds in noisy environments like a classroom, right? So, and this can be kind of months to years even after the hearing loss has been kind of removed and normal hearing restored. So there's some persistent change, we think, in the central nervous system that causes these kids to kind of hear the world differently. So you can have kids that are sitting in the classroom and they say, I just can't really hear the teacher. Even though they're each, when they have their hearing tested, we call that an audiogram. You've probably all done that. You put the headphones on, you listen for the beeps. Each ear is perfectly normal, right? So it's not an audibility problem, but you put these kids in a kind of noisy, complex environment and they just can't really focus their attention. So these are sort of the who cares questions, why we want to look at these questions. And sort of following, so how do we do that in a laboratory setting? We need to do this in a very controlled way. There are clinical groups that are trying to look longitudinally in kids, but you often don't really know. There's no real control. You can't understand day-to-day kind of what the hearing loss is. So they're all kind of what we call retrospective studies, trying to correlate what might have happened with some current kind of behavioral outcome. But we wanted to be a little more scientific about this, where we could really control the sensory environment in an animal model, not unlike putting the patch on in the classic Hubel and Wiesel studies in the cat. So instead of a patch, we essentially patch an ear, plug an ear, and we do this by placing, I don't have that slide, essentially custom molded silicone earplugs into, in this case, guinea pigs, into one ear, so we have a real big asymmetric hearing, and we do this for 8 weeks.
So they're used to the plug.
They're used to the plug. Yeah,
Okay.
they're sort of custom molded. They're not foam earplugs, so we basically use, and I don't have that slide, but we use a silicone injection material. It's the same kind of stuff that if you went to have a custom made hearing aid, the clinicians would basically inject this goo into your ear and make a custom mold. So it basically really nicely fits in there. It doesn't hurt them, and they never try to scratch it out.
Right.
So,
but you leave it in for 8 weeks.
we leave it in for 8 weeks,
and they get used to it.
Yeah.
Ah.
Yep. So they don't know it's there. They're just going about their normal guinea pig business for 8 weeks. And I'll get to why it's 8 weeks in a second.
Okay.
But people always ask us, because in my lab we use some unusual beasts. It's not the usual rats and mice kind of thing, although we do use those for some purposes. We use guinea pigs and chinchilla for a couple of reasons. One is that they're precocious, so they're actually born, not unlike human, with with pretty functional vision and audition. So they can kind of see and hear. So much of the early development of the auditory system takes place in utero, right? So in human, we start to hear at about 28 weeks of gestation. And it's kind of not that long in the chinchilla and guinea pig, but they begin to hear it in utero. And what's cut off here is one of my former graduate students, Heath Jones, basically showed in the chinchilla that, essentially at birth, the peripheral auditory system, kind of the cochlea and stuff like that, is basically adult-like. So the external ear is sending kind of normal, good acoustic information to the central nervous system, basically, at day one. And it's a similar thing in the guinea pig. The other is that these two species, guinea pig and chinchilla, actually have ranges of hearing that are essentially identical to the human. So they have good low frequency hearing, which species like mice and rats don't have. They don't hear low frequencies, and kind of moderate high frequency hearing. So if you look here at the so-called audiogram, kind of the range of frequencies that we can hear, you can see that chinchilla, for example, matches almost exactly the human. And what this means is that these animals use sort of all of the acoustical cues, these interval time difference cues and interval level difference cues, in a way similar to humans. So we've done behavioral work in these animals, and other labs have too, to demonstrate that they use the cues in the same way we do. So we think it's a better model for the kind of translational work that we're trying to do. So when you guys see that, that's sort of the rationale for why we use these unusual beasts. Okay, so again, we put the earplug in for 8 weeks, and the reason we do that is because these are some measurements that Kelsey did in a kind of a big litter of guinea pigs. Essentially, by about 8 weeks, the head is fully grown in the guinea pig, right? So that really approximates this range in the humans where we are where we sort of think that the sensitive or critical period needs to be, right? We think that the nervous system needs to accommodate for this essentially doubling in the size of the head, which changes the acoustical information. So that's our rationale for plugging the ears for 8 weeks.
Dan, do you play music and move it around, or do you just have it be the white noise during those 8 weeks?
We don't really do anything. It's just their normal kind of animal care environment. They do have they have this I think they call it Thunderdome or something. So that the animal care staff, it's like a kid swimming pool that the animals get to play in on a daily basis. So they chat and they just go about their normal business. So, but that is a good question. It's another question of whether some of the things I show, some of the detriments that I show, can we train, provide some additional input, sensory input, or enrichment that might help overcome that. And that would be sort of the next stage of experiments. Here we just wanted to have passive, as they go about their daily life, just like a kid would go about their kind of daily life without any other sort of intervention. Do these animals experience some deficit via this earplug?
Don't you have to change the earplugs because if the head grows, the ears grow? Yep,
great question. Yep. So Kelsey was a real trooper, and Alex as well on these. Every few days, particularly early when the head is growing pretty rapidly, the earplugs fall out. So between the animal care staff, Kelsey and Alex, me and some of the others, we would check several times a day, even on the weekends. And if the earplug fell out, you would just put another one in. And yeah, that's a great question. But I've got a slide later where we have essentially an earplug retention score. Basically, we sort of have an idea of over that 8 weeks, kind of what's the percentage of time that we were confident that there was an earplug in place. And for all of the animals I talk about, it's basically about 85% of the time. So it's in there most of the time. So this slide was misplaced. So this is basically the silicone impression material, and you can see a kind of a custom earplug here. It fits really nicely into the ear canal, and it can't really ask the animal. It doesn't seem to bother them, right? They're not trying to scratch it out. They just kind of go about their normal business.
They don't say what.
What? I'll show you in a moment. They do say what, and that's what this shows here. So one of the things that, or one of the ways that we can assess spatial hearing pretty rapidly is to measure something called the minimum audible angle. And essentially, this is the smallest change in the location of a sound. So if I played you two sounds at two different locations, what would be the smallest angle that you could just detect? In adults, we are fantastic at this. We can detect about 1 degree or less. And just to give you some idea what that is, if you held your arm out, if right in front of you, if you moved a sound from one side of your thumbnail to the other side of your thumbnail,
Wow.
you could detect that. We're amazingly good at, I think barn owls might be the only species on Earth that are rival what we can do. Yeah, it kind of, if I could just digress, it's kind of strange, like why do we have that level of acuity, right? Because, we're not putting food on the table, using our sound localization like a barn owl would. And really, we think we use it for these kind of social situations where there's lots of noise, but we really, we need to, very importantly, be able to focus down and listen to instructions or things like that. So that may be one reason why we have this great precision. But one interesting thing that's been known in the field for quite some time is that this capability takes a long time to develop relative to other kind of basic auditory sensory tasks. So this takes, again, 5 to 6 years to get up to kind of somewhat adult levels of capabilities. Other tasks like frequency discrimination or intensity discrimination are basically adult like pretty early, right, within the first few months. So the question had always been, well, what's the deal here, and why does this last so long? And again, we think it's this period of plasticity that the system is compensating for the change in acoustic information due to the growth of the head. And so we can, we want to measure what this is in our animals. So we know that there's this developmental trajectory in normal hearing kids, kids that have had this conductive hearing loss due to fluid accumulation in the ear due to ear infections. They're often really poor at this task. Kind of, they perform it as well as kind of a really young infant, even though they can hear perfectly normal. So it's something in the central nervous system, not because they can't hear the sound. And so we wanted to ensure that our animal model sort of exhibited this similar trend. And it's actually very difficult to do sound localization work in these rodent species. You're essentially trying to ask them, can you tell the difference between two sounds presented at two different locations? The classic way to do this is to use a task that we would call operant conditioning, where you basically try to train the animal, give them a food reward to do the task. That's actually very difficult in rodents to do that. They just don't really understand what they're trying to do. And because rodents aren't predator, they're actually food, right? They're prey.
Mhm.
They don't inquisitively look at sound sources. It's not like a cat or a dog where you play a sound and they want to look at it and interrogate it. These guys don't do that. So one of the strategies we used here is to test this is to use elements of what we call acoustic startle. So if I play a very brief but loud sound to you, you'll jump, you'll startle. And animals will do that too. So if I play a loud sound here, this is actually our testing apparatus. I've got this array of loudspeakers here. The animal is seated in this cage here, kind of facing this bank of speakers. And above here, it's kind of hard to see, there's another loudspeaker that plays this startle sound. So if we just play that startle sound just kind of at some random time, the animal will jump, and we can record kind of the magnitude of that startle, that jumpiness, by what we call an accelerometer placed on the bottom of the cage. And this shows that startle here. So when we play that sound, you can see that the animal jumps and the cage kind of moves.
Mm.
But what we know from this, again, it's called acoustic startle, is that if you change something in the sensory environment, the animal actually startles less, right? So people think that this is it's like a rabbit out there, that'll just kind of, it doesn't want to startle, right? So it prepares itself kind of for this loud sound. So here we sort of leverage this kind of natural reflexibility. We present a sound at one location, so it's kind of going shh, 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 shh. And then we swap it to another location. And then very shortly after that swap, we play that startle stimulus. So the notion here is if the animal can detect this change in speaker location, it'll startle less. And indeed it does. So This red line is a trial in which we swapped the speaker relative to the blue trial when we didn't, right? It just kind of stayed at the same location. And sort of using this technique, we can basically change the angle that's subtended, so that they're very close. And it turns out when they're very close, the animal can't tell, right? It startles sort of the same, even though we changed the location, it startles similarly to as if we didn't change the location at all, which we sort of imply that the animal couldn't detect the change. But if we make the swap very large, the animal can detect that. So essentially we can carve out a function here. At very small angles of speaker swap, the animal can't detect the change. At these much larger ones, it can. And we can sort of put what we call a threshold value. So what is the change in speaker location that the animal could sort of statistically detect reliably? And in a normal hearing group of animals, they could detect about 7.5.
Did it go to sleep or?
I don't think so.
Huh.
No, I shouldn't have done that. What the heck?
There we go.
There.
All right, I'll keep talking while Megan adjusts. Thank you. Okay, so we can put a threshold. So essentially, normal hearing guinea pigs can detect a pretty small change. That if they're, of course, worse than we are as humans because their head is smaller, but they can detect about a 7.5 to 15 degree of change, which is pretty good for these small animals. But after this, the kind of litter mates that we put the earplug in for 8 weeks, I should say we take the earplug out. We have a variety of ways that I'll talk about in a little bit that we can assess whether the hearing is normal in the two ears, and the hearing is normal. The earplug hasn't done anything to what we call the peripheral auditory system. So that ear that had the earplug can hear perfectly normally once we take the earplug out, right? Because it's essentially just a mechanical hearing loss. It's just a plug in. So after this period of earplugging, these animals are much worse at discriminating the spatial location by about 2 to 4 times worse. So they require about 30 to 45 degrees of change, right? A big change. It's a little complicated here, but even though we're changing, because it'll make some sense when I talk about the physiology here in a moment. So when we change the spatial location of sounds, of course, we're changing the acoustical cues, these interval time difference cues and intensity or level difference cues at the two ears. So we think it's those cues that the animal is using, particularly for this task where we are sort of isolating their ability to use just this intraoral level difference cue. So we can essentially measure what those cues are by placing small microphones near the eardrums on the two sides of the animal and figuring out how big of change in this intraoral level difference cue resulted from changing these speaker locations by either the amount that was in the control, 7.5 to 15, or this larger amount, 30 to 45. And essentially, in order for the animals to do this task, if they're using the intraoral level difference cue, which we think we've forced them to use for a variety of reasons, that would require them to be able to discriminate this intraoral level difference cue, kind of the difference at the two ears of about 1.5 to 3 dB decibels in controls, and again, about 2 to 4 times worse than that in the conductive hearing loss. Okay, so these are the things that are going to allow us to then look inside the brain and ask questions about, how well can the neurons discriminate these kind of values? Okay, and the question is, well, where actually should we look in the nervous system for these changes, right? We have these big behavioral changes that occurred. Where might we look? And so the interesting thing is this startle task. We, or the field, sort of knows a lot about the neural circuits in the nervous system that mediate this. And so the normal acoustic startle pathway is a pretty short neuron arc. So you startle really rapidly, right, when you hear a sound. And that's because there's not very many neurons or synapses between hearing the sound and producing this motor response. But it turns out that motor response can be modulated by this other circuit. And I won't go through this whole thing, but one critical group of neurons in this is the so-called inferior colliculus. And the inferior colliculus is a sort of a really important auditory system neural structure that receives inputs from all these lower brainstem. I won't go through this mess of stuff here. I like this figure because it just shows kind of how interesting and messy the auditory system is. I'll just say, we have the external ear, the cochlea, that's basically processing the incoming sound and then sending neural information to these other structures that are extracting the acoustical cues to sound location. One of these groups of neurons, called the lateral superior olive, receive excitatory input from the near ear and inhibitory input from the opposite ear. So essentially these neurons, we think, are subtracting the sound, the neural representation of sound intensity of the two ears, right? This is the circuit that you would design if I asked you to design a robot to tell me where a sound would come from. Well, the first thing you do is just try to compare the intensities of the sound at the two ears by subtracting them, and that's exactly what these neurons do. That information is then sent to the inferior colliculus on the opposite side of the head. So neurons in the inferior colliculus, many of the neurons are sensitive to the interval-level difference cue to sound location. And we wanted to study how do those neurons then encode the cues to these cues to sound location in both the normal and the hearing impaired animals. So the way we do this in the laboratory, we deeply anesthetize the animals, and we isolate and record from neurons in the inferior colliculus using very fine electrodes. So these electrodes, the tips of which are smaller than the size of a human hair. They're really tiny. And we place those into the inferior colliculus to basically record the neural activity of populations of neurons in this structure. This is essentially what we find for many of these higher frequency sensitive neurons. So what's plotted here on the y-axis is essentially the number of action potentials that a neuron fires per second, or what we call spikes per second, as a function of the intensity, the difference of intensity of sound at the two ears, this interval level difference cue, right? And these neurons are very good at encoding this kind of spatial cue, this interval level difference cue. So if we have a sound that's louder at what we call the contralateral ear, but it doesn't matter, that's louder at one ear relative to the other ear, the neuron fires a lot of action potential. So it's really active. But then as we sort of move the sound location so that now the intensities that the two ears have swapped, the neuron can be completely silenced, right? It's completely inhibited. So the beauty of these neurons is that they provide what we call a neural code for the horizontal location of sounds, right? So as we move a sound from one side to the other, the neurons respond a lot or don't respond at all. So you get this nice kind of sigmoidal relationship between the number of action potentials and this cue to sound location. So we wanted to study populations of these neurons in both normal hearing and the animals that were hearing impaired. I'm not going to go through all of this. Essentially, we're going to ask the question. So you might say, well, who cares? You have these neurons that have this interesting shape. It's encoding sound location. But we wanted to ask more of these neurons. So we developed some computational techniques to basically, you can imagine, well, there must be some other neurons at the next stage that are listening to these neurons in order to make some guesstimate about where the sound was or some appropriate behavioral response. So we sort of simulated what would be the significant information that the next stage would receive from these neurons. And what we essentially asked was, well, how small of a change of this acoustical cue, this interval level difference cue, could these neurons detect based on the fact that you have this shape of the curve, so basically the slope of this curve. So as we change this this acoustical cue, these distributions of neural responses are changing. So essentially we're doing a statistical analysis of the spiking by these neurons, and we're looking for the situation where, again, another neuron looking at this could confidently identify that this cue had actually changed its value. Okay? So essentially we're comparing two distributions, not unlike a t-test or something like that. So we've just developed kind of fancier t-tests in a way. Again, I'm not going to go through this, but one thing we can do with this sort of complicated math is to, again, find that what is the smallest value that the neuron can just detect. And so we do that, and I'm just going to summarize that. And in order, the whole reason we do that is so we can compare that to the behavioral capabilities, right? So we can essentially ask the neuron, you tell us how small of an angle you can just detect, and we're going to compare that to the behavioral measurement of how small of an angle. So, but we're going to cast that again in terms of this acoustical cue that supports this interval level difference cue. So what's shown here in this blue bar is essentially the behavioral capabilities of the animals, normal hearing animals, to detect a change in location. So they can detect this range. And this is what the neurons can do. So you see that there's a really nice, in the field we need to be sort of careful. We're not saying that these neurons predict what the behavior is, but they're the values that these neurons are able to support match sort of the behavioral capabilities. Here's the behavioral capabilities after animals have been reared with the earplug for 8 weeks. So they're, again, about 2 to 4 times worse. And you see the neurons in those animals are really different. So there are some neurons that are still pretty good. And we think that these neurons support, again, the audibility in the 2 ears is good. They can still hear. They're not deaf by any means. They're just really impaired in this spatial hearing task. But you do see that there are many neurons that are really bad. And you just never find those in kind of the normal control animals. So we think this essentially provides actually the first kind of neural basis or explanation, if you will, for why these animals, and the hope is for why these kids that had these problems experience these issues with spatial hearing. So we think it's a change down in the brainstem. And this sort of gets back, so the who cares question, right? So again, we think, we know clinically that kids that have had sort of chronic hearing loss due to ear infections have these really persistent problems, speech and language, localizing sounds. And so how might this play out? Why do they have difficulty in sort of, not cocktail party, but kind of classroom settings, things like that, where there's noisy sources? So here's our sort of explanation for that. So, you've got the kid with the hearing problems, the troublemaker kid back here. I don't know why he's in the back of the class, but that's where Kelsey put him. Try to listen to the teacher, right? And of course, there's other students, other sound sources that are competing, right, for the teacher's voice, right? A sort of normal hearing kid has this really narrow, we almost think of it as a funnel, right? So we have this spatial hearing capability, and we can sort of aim that funnel. This is sort of the example in the cocktail party, right? I can pretend I'm listening to Rob, but really I'm listening to Robbie or Megan's conversation right behind
They're
me.
more interesting.
Yeah. So we have this ability to kind of focus our auditory attention, in different directions. This is where the spatial aspect comes in. So, we can aim our funnel of attention, and the idea here is that, we can't resolve anything kind of inside that funnel. So whatever falls inside that funnel, we sort of process that. So what we want is a really narrow funnel, to aim that at the speech of the teacher, so we can receive that information and basically kind of squelch out the other distracting sounds. But as we just demonstrated, and what we think is happening in these kids, that this period of hearing loss, this temporary hearing loss, is just disrupting the normal kind of experience-dependent tweaking up of this funnel, and it remains really large, right? So now not only does the speech that we want to hear fall in that funnel, so does these other speech sounds. So you just get this mixing of inputs, and we think that there's no way for the nervous system to tease these inputs apart. They're kind of all merged together, and as a consequence, the speech perception of this kid trying to listen to the teacher is sort of muddled. Again, even though audibility is perfectly normal at the 2 years. So that's a couple of the reasons why we're interested in studying this. And as I mentioned, it's very difficult to assess the hearing problems in these kids. We often, we have this sort of kind of throwaway term that's used in the field called central auditory processing disorder. These central auditory processing disorders are characterized by kind of auditory difficulties, things that are modulated temporally, like speech, sound localization, detection of sound in competing environments. And these are very difficult things to assess clinically, okay? Because the kids are often referred. They're often referred because they think the kid has ADD or ADHD, and it often they just have these really subtle hearing problems. So we'd really like to come up with some way to objectively assess this in the clinic. I don't have time to talk about that, but it's sort of one of the sort of core motivating reasons why we're looking at these issues. And I would just say that, and perhaps next year when I can update you guys, we've been looking at basically objective measures by which we can assess this in the clinic. One of the ways we can make what we call objective, so by objective we mean sort of things you can measure. They're not subjective, like asking you whether you can hear a sound, because those are really difficult to do in kids. So we want these objective techniques, like you can't just ask the baby how their cocktail party skills are, right? You need some objective way. And one way we can do that is to measure what we call auditory brainstem responses. So we hook up these non-invasive surface electrodes. So these are basically little patches that you can stick on the forehead. It's sort of the exact same idea of measuring kind of your heart. The EKG, you put some electrical leads on the surface and you can pick up the electrical activity of the heart to assess whether there's a problem or not. This is the same idea here. We put these little contacts on the surface of the scalp. We play sounds to the 2 ears. You can see these little insert earphones. And what we record, so unlike the EKG where the heart is providing, like Kathy has probably told you or will tell you about the electrical activity in the heart, we sort of have to provide the stimulus. So we present sounds to the 2 ears. But what we measure at the surface of the scalp are these evoked potentials. So we have these kind of peaks. This is basically an electrical waveform that we can record. And you see there's these kind of discrete peaks. These peaks represent synchronized activity of populations of neurons in kind of well-defined stations in the ascending auditory system. So like wave 1 comes from the auditory nerve. Wave 2, which at least the way we record it, we don't see it, comes from the cochlear nucleus. Kind of 3 and 4 come from these center parts. And we've developed some techniques where we think we can use properties of particularly this wave 4 to diagnose spatial hearing problems objectively. Right? We don't have to ask these kids anything. We can just hook them up, measure them, and at least we'll have some understanding whether there's some fundamental problem with this brainstem circuitry as the explanation for why they claim they can't hear well in a classroom setting. We think if these brainstem properties are normal, then they might be candidates for other things like ADD, ADHD, things like that are more higher level, more higher cognitive problems. So we're really interested in having techniques where we can basically rule in or out really peripheral fundamental hearing capabilities. And then our idea is that by knowing that, we can then, or at least the clinicians can then, by knowing kind of where the problem is, then develop strategies, rehabilitation techniques, things like that sort of are aimed at those. So I'll stop there. I think that's enough for you guys. Inundated you with interesting things about how we're thinking about plasticity in the auditory system, the role of experience, the role of abnormal experience. So, having this capability to have a nervous system that can learn pretty rapidly during development is a good thing, but it can also be a bad thing, right? If you have some deficit in the inputs, right, the brain doesn't know that, it's got a hearing loss in one ear, it's going to try to optimize to that. So now when the hearing loss goes away, the brain, as we think, has been sort of, optimizing the wrong information. Now the right information comes in and it has no idea what to do. So you end up with these kind of perceptual deficits that can persist for a long period of time. So thank you guys. Again, thank you for your outstanding work to help support. Without you guys, it'd be much more difficult to get these things off the ground and up and sustained as well.
Dan, is there any evidence of being able to reverse maybe not necessarily, you're trying to figure that out now in this, but in other areas of of cognitive processing?
Well, certainly the classic example in the visual system, right, is wearing the patch, right? So
that's actually a cognitive treatment, not a.
Well, we think that's a sensory treatment that. So there's essentially what we think in the visual system and also here in the auditory system is that in order for these kind of really fundamental brainstem circuits to wire themselves up is that you need sort of a healthy competition between the inputs from either the two eyes or the two ears. And if one of those is disrupted, then the input from the kind of the normal seeing or hearing side sort of takes over, right? It just kind of co-ops all of the neural space. And so there's really nothing left, even though you restore vision, you restore audition, there's really no chance for that to overcome that. So the thought is that sort of Hubel and Wiesel came up with that's now sort of routine in clinical practice is that you basically give the weaker side a fighting chance by, for example, in the visual system, putting a patch over the stronger eye. Right? So now you're allowing the weaker eye to, you're sort of shifting the balance, right? Making it a more fair competition. And that works pretty well.
So if you plugged the good ear.
That's what we're thinking.
Yeah, okay.
We haven't done any of that, but that would be the hypothesis that you basically, using these objectives, again, you can't ask a baby how the hearing is, but, we think we have these ability. We've got, fantastic evidence from, these animals that we plug that we can, essentially without doing any behavior or looking at the physiology, we can measure these auditory brainstem responses, and we can predict which animals were impaired and which weren't.
Right.
Again, a lot like a baby, we can't ask the animals, right? But we have these tests. So the hope is that we can use that technique to try this out. So we know that rearing an animal with 8 weeks of hearing loss in one ear, really messes them up.
Mm-hmm.
If we now put it in the other ear, can we track their progress using this objective metric?
Yeah.
With the understanding that perhaps once this auditory brainstem response metric is back into kind of the normal range, we would hypothesize that the behavior would also be normal. So that's sort of the next level of experiment we're going to do to kind of demonstrate the clinical feasibility of doing something like this.
Wow.
I don't know how you're going to get a kid to run it. So, my daughter would never wear she tore the patch off all the time. There's other techniques where you can put the atropine, when you go and have your eyes tested, they dilate your eyes. So you can also do that. You can basically dilate the good eye or the stronger eye. So you're providing kind of a weaker input in that way to one eye relative to the other eye. And that seems to be just as effective. And the kids tolerate that. And need to run around with a patch on their eye.
Is the expectation that with an older animal that's already past that period of plasticity, that it would then take longer?
Yeah. So I didn't mention that. So so actually Alex Ferber did these same kind of experiments, but in kind of juvenile, kind of the animals post normal hearing animals. He would put the earplug in at about 8 weeks and leave it for 8 weeks. And those animals showed virtually no deficit in their spatial hearing, and also this underlying electrophysiological response. So, kind of those two things, that there's some period of plasticity early, somewhere in that 8 week period, but not after that. But there are some changes. We know from the clinical literature. I didn't really mention this, but there are other. So I mentioned this central auditory processing disorder. It's sort of most commonly diagnosed in kids because that's most of the clinicians see for this problem. But there are other groups of people that experience these same types of symptoms. As we age, we sort of lose our hearing. It's called presbycusis. But there's a new class of kind of hearing loss called hidden hearing loss, where essentially we have pretty normal audibility. This is pretty common now in aging populations that they complain, I go to a restaurant, I can't have a conversation. They have their hearing tested, and it's pretty normal. So audibility, again, is not the problem. We think that there's some central nervous system problems. So actually, Occam Klug and I recently put in a multi-PIR one to kind of use some of these techniques that we've applied in kind of early infancy and development, but kind of on the other end of the aging spectrum where we think there's sort of similar issues. The issues with aging is that we think over time we start to lose neural inputs. It doesn't really affect audibility, but essentially the strength of inputs to the central nervous system is reduced as we age. And that's not too unlike reducing those inputs by putting an earplug or having a hearing loss due to an ear infection, right? You're reducing the inputs to the central nervous system. The nervous system is going to try to tweak itself up to compensate for that. And we think that's a similar thing that's going on in aging. Other things like neurodegenerative diseases like multiple sclerosis. The other thing, the blast exposure, some of the things we're working on with the Department of Defense, TBI, things like that. These subjects experience similar problems of central auditory processing disorder. Again, the audibility is not the problem, but something is happening in what we call the central nervous system, right? So kind of beyond or more central than just the cochlea and the hair cells and things like that. So the hope is some of these techniques that we develop will be able to kind of move them into a lot of different areas, just not the developmental realm.
Any other questions for Dan?
Yeah.
So when the animals are born or humans, they say that the structure of the ear is already in place and it grows and expands as the head doubles in size, or is it something that's growing and developing during that plasticity?
Well, in human, and we think in these animals, the cochlea, kind of the little snail-shaped structure that is kind of sensing the vibrations, that's adult-like. It's the same in the human. Actually, your eyeballs are adult size when you're born as well, which is kind of crazy, right? And they're essentially just moving further apart. And the cochlea is the same, right? It's just, you're essentially moving the ears further apart. There are some morphological changes in like the size of the external ear, the size of the head. No, that's changing the acoustic information. That's the basis for which we think that there needs to be some plasticity to adjust for. Essentially the head is growing so fast as a young infant that almost every day a sound at a given, let's say 45 degrees off to your left, has a different set of information that specifies that location, right? But there's no real evidence that kind of developing animals or developing kids, they're not sort of mislocalizing sounds, right? They sort of know where that sound is. So we think that there's this ongoing calibration that's kind of keeping your sensory world stable, even though your head is growing and the sensory input is changing. But then you could disrupt that, right, by putting an earplug or fluid in the middle ear, or other types of hearing impairment.
And then one more question. So for kids who have CAPD, is there evidence that they have balance issues as well, or is it just?
Well, that's a good question. I don't know that. Yeah, good question. We know a lot of these things are kind of intimately tied to balance. So people that have hearing problems tend to fall more than people that don't. It's kind of a new thing with all the aging population we have, which is kind of an interesting thing that there might be this, we don't know, but it's, the balance system is right in there with, what did I do with the ear? Oh, anyway, right, the vestibular apparatus is basically, right next to the auditory system. Actually, the fluids that comprise the cochlea are contiguous with the fluids that comprise the vestibular apparatus. The mechanisms with these little hair cells, these hair bundles, they sort of have the same functional properties. So, I think they may be more intimately tied than we often think.
So Dan, I was in a clinical trial where they were trying to change your food preference by subliminal messaging,
Yep.
Flashing before your eyes during an MRI.
Oh.
And I never found out if I was in the placebo
Yep.
group or not, but I don't think I was because I liked vegetables a lot more after that.
Yep.
But the thing that is bizarre to me is that your eyes cannot see fast enough, but your brain can take in the message.
Yeah, for sure.
So I'm wondering if there's sort of that same sort of thing with your ears where, okay, well, we don't have this spatial thing, but maybe there's a way that we can subliminally go to the brain through the ears too.
Yeah, no doubt. We sense so much stuff, right? Like you can't get computers to do, even as fast as things are, we can't get a computer to kind of process sound in a cocktail party environment, right? We do it pretty, you don't even really have to think about it. And so one of the ways that we kind of get around that, kind of this barrage of sensory input, is that we're pretty selective, or the brain is pretty selective about what kind of reaches consciousness and perception.
Mm-hmm.
With the understanding, maybe we learn those things to kind of get rid of things that don't really mean anything. And there are things, so there are other routes of information in the auditory system. So kind of the classic Pavlovian fear conditioning, right? So you pair a shock with a sound, and then or food with something, and then all you have to do is give the stimulus. You don't have to give the other, but and that all goes through a pretty low level of the auditory system. So from this inferior colliculus, there are projections to the amygdala, kind of our fear and emotion processing center. And people have often speculated that, the sort of sixth sense that people have, or, the hair stands up on the back of your neck sort of thing, you're getting input via those other circuits, right? You're not really aware of what that is, but there's these other mechanisms that are still sensing the environment, perhaps preparing you for, different actions or things like that, even though you may not need those to kind of rise to conscious perception in order for that to be useful. And there may be ways to leverage that, kind of tricking the auditory system to kind of receive information, or using that to kind of train, retrain the system. A lot of things we don't know.
Really
It's
cool.
kind of the more we dig in, the less we know, I think.
Well, we should draw to a close, but thank you, Dan.
Great question.